Okay, so good afternoon, everybody, and welcome to our plenary panel. So myself and Ben, who is from the University of Bath, are going to be co-chairing this session today. So you'll hear from both of us as we go through. So as with the other sessions, if you've got any questions, please pop them into the Discord channel under the plenary hashtag, and we will do our best to get through as many of those as we can. So we're going to move on to introducing our panel. So I'm going to kick things off by introducing Professor Sam Sheppard. So for those of you that don't know, Professor Sheppard is a Professor and Director of Bioinformatics at the Milner Centre for Evolution at the University of Bath, and he is also the co-PI of the MRC Cloud Infrastructure for Microbial Bioinformatics, which is the UK's largest cloud-based system for the analysis of microbial genomes. Research in Sam's group centres on the use of genetics, genomics and phenotypic studies to address complex questions in the ecology, epidemiology and evolutions of microbes. Their most recent interests focus upon comparative genomic analysis to describe the core and flexible genomes of pathogenic bacteria, including Campylobacter, Staphylococcus, Helicobacter, and E. coli. So welcome, Sam. We are absolutely delighted that you are here with us today. So especially as for those of you that don't know, Sam actually injured his knee on Wednesday, has been in and out of hospital for the last 2 days, and in true dedication to science and Campylobacter, he's joined us this afternoon. So thank you very much, Sam, for being here.
It's a pleasure to be here, Lisa.
Okay, so I also have the great privilege of introducing Dr. Arnoud van Vliet. So Arnoud obtained his PhD in 1995 from the Veterinary Microbiology Department at Utrecht University. For his postdoc, he moved to the Department of Genetics at the University of Leicester, working on gene regulation and virulence of Campylobacter jejuni. So Arnoud has been working with Campylobacter for a very long time. In 1999, he obtained a personal fellowship from the Dutch Organisation for Scientific Research to work on metal metabolism gene regulation in Helicobacter pylori in the Netherlands. In 2007, he took up a position as a research leader at the Institute of Food Research in Norwich, where he led a research group focusing on Campylobacter and Listeria, combining microbiology, molecular biology, genomics, and other omics with bioinformatic technologies to understand the process allowing these bacteria to survive in the food chain and cause illness. Arnoud joined the Surrey Veterinary School as a senior lecturer in veterinary microbiology in October 2006. Thank you, Arnoud, for agreeing to be part of our panel today. We're really happy you could join us. I know I've been promising sparks are going to fly, and Arnoud, through his background, has already achieved that promise because the sparks are flying behind him.
Well, thanks, Lisa, and it is my pleasure to introduce Professor Christine Zamanski, who is a Canadian at the University of Georgia. She's a chair in bacterial glycomics and was among the first to demonstrate that bacteria can N-glycosylate proteins and now exploits these systems to make glycoconjugate vaccines. And she sometimes does this with Professor Brendan Wren from the London School of Hygiene and Tropical Medicine, where he's a professor in microbial pathogenesis. Brendan was, along with Julian Parker, was one of the first to sequence the Campylobacter jejuni genome all the way back in 2000. And Brendan and Christine are definitely among the world leaders for studying glycosylation and glycostructures and exploiting these interesting structures for developing vaccines for veterinary and medical use. So I think, Lisa, are you ready to ask the first question? And then we'll try and bring in as much from Discord as we can.
Yes, of course. So this was supposed to be a nice, easy question, but of course, for Sam, this might not be as easy because it's going to focus on the last 2 days of the conference. So I'm going to tweak the question slightly. So what talks or recent research that you have seen has excited you over the last few days or months? So I'm going to start with Arnoud, please.
Oh, on the spot straight away. It's a good question. I mean, there is a lot of progress being made with the different groups and the different fields that are there. And of course, my interest nowadays really goes towards the genomics and the things that are happening there. And in a way, it's both being excited about all the new knowledge we get from the genomics, but also realizing how little we still know more than 20 years after having a genome sequence, and we really think like, yes, we're going to answer those questions. And we had that first genome sequence, and in a way we were really disappointed because there's nothing there that actually explained anything from what we didn't know before. But I think what excites me the most is the explosion of possibilities that the technology is really bringing now, and that everybody can do it. So it's not a selected few anymore.
Thank you. Brendan, can I ask the same question to you, please?
Yes, I'd actually like to point the audience to a paper I just read recently that hasn't been referred to in the meeting, and that's a publication, Plus Pathogens, this month from Jos van Puten's group in Utrecht, and it's to do with virulence, and it's still one of the big mysteries that we don't really understand. You know, why Campylobacter are so virulent in humans and they're not in avians, etc. And in this paper, they showed that heptose phosphatases were responsible for certainly activating a large inflammatory response. So the data looked quite convincing, and that particular paper not being mentioned in this meeting. And I think the other area that in the future, and I'm sure we'll touch on it, is despite some of my comments earlier, I think that there is a real need for vaccines, particularly in poultry, for Campylobacter. And Christine and myself have been working on this, and I think that will make a difference in terms of what's going on, because as Frida showed, the numbers of Campylobacter are still increasing in the UK despite COVID, when you might expect them to drop. And frankly, it's a national disgrace that we still accept this, because Campylobacter is a pandemic. It has been for a long time. And you can get severe disease, as we all know. So we need to highlight that this is still an ongoing problem. But along with what Arnoud said, I think that with technology and some of the other things I've heard at the meeting, there are real opportunities to really try to reduce the amount of Campylobacter in the food chain.
Thank you. Yes, the data showing that people still getting ill in the pandemic is really interesting, because perhaps it's the first evidence that people are poisoning themselves in their home rather than blaming the takeaway or their friends cooking at a barbecue. So I think that's really interesting. So Christine, can I have your view on what has excited you recently, please?
I'm excited to hear about Campylobacter and to join all my colleagues online, but specifically, we heard a lot of talks about phase variation, and that's something that still bothers me in a lot of the publications that I see, because we make a mutant in Campylobacter, we isolate a single colony and we compare it to the wild type, and we think that mutation has all those phenotypes, and we don't think that single colony could have actually had all other sorts of variations that have nothing to do with that mutation that was made. And so that's something I think that's riddled the Campylobacter literature a lot, with a lot of inaccurate conclusions, and something that I think especially the new generation needs to be aware of is, Brendan, I think, called it once Campylobacter, or a moving target, or some terms that Brendan liked to use. And I think we really need to emphasize that that's the case, that we really need to be aware of the fact that things are changing. But the other things that I've been very excited about are all the publications and studies that have been coming out lately on the metabolic capabilities of Campylobacters. I think that obviously another area that we're very excited about and working in. So I really enjoyed listening to those talks. The conversations that came out on what really is a virulent Campylobacter, and then Brendan saying, do any of us feel comfortable in ingesting any Campylobacter, whether it has a certain structure or components that we label as a virulence factor or not. That's something else I think we need to be thinking about. And then the other aspect that really came through, I think, in the talks over the last few days is the fact that Campylobacter isn't necessarily just a commensal in chickens. Maybe it's more of an opportunist, and we do see spread, and we do see some chickens that appear less healthy, and that vaccines should also keep that in mind, that we could potentially improve poultry health by vaccination. And so those are all things that I thought were emphasized many times throughout the last few days.
Thank you. And finally, Sam, what's been exciting you in the world of Campy recently?
Okay, so obviously I missed quite a lot of the talks over the last couple of days, but I've caught up with a few already online, and I won't say we've made a short list because there's a prize, of course. So I'll announce some a little bit later on. But I want to say that after a time of isolation and all the awful things that come with that, I'm delighted to be here in this company and see my old Campylobacter chums. And I suppose what I think, Lisa, to start with is having been to lots of these meetings over the years, like many other people, I sort of remember when there was loads of conversation about nomenclature, or should we use MLST, or should we use some other typing scheme, or all of this stuff was debated for many years. And of course, as Arnoud says, now we can, with the democratization of genome sequencing has come, this kind of ability for everyone to start thinking about Campylobacter as the genus as a whole. And so what's really important to me is that we don't think about individual lab strains, but that we think about the difference between strains as well. And that still gets me going now, because we can, in loads of even in some of the talks that I've already seen, and obviously the ones from my group that I knew about, we can see that there are host-associated lineages. We can see that there are lineages that are more likely to be antimicrobial resistant. We can see lineages that, we know about lineages that are a bit more virulent than others, or certain ones that are very virulent, and all this sort of thing. So I think the kind of population genomics, understanding the species or the genus as a whole, really gets me going. And within that, just sort of building on, I love the phase variation stuff, and obviously we don't understand virulence very well, and all of these really important things. I think we also have to just remind ourselves that we still don't really understand horizontal gene transfer brilliantly. It's obviously a major force in the evolution of Campylobacter in lots of different ways, but we still, it's not really clear sometimes how much there is, what effect it has, how you balance the benefit of acquiring something new against the potential downside of disrupting an already co-adapted genome. These sort of genetics questions are really obvious in Campylobacter and quite important. Oh, Brendan's saying something, but he's muted. Oh, he wasn't saying it to me. He was perhaps. Anyway.
I was telling my wife to go away.
Oh, were you? Okay. All right. Well, there's one more thing I just wanted to say as well, that's just from the talks that I've caught up with, and also this relates to what Brendan and Christine said, and Arno just said already. But one thing that I really think is great is that we're starting now to talk about Campylobacter in low and middle income countries. So for many years, most of the focus, I think I'm right in saying this, most of the focus has been on Europe or North America, probably. And when you've got sort of chronic diarrheal disease, that's not going to kill you, but it's really bad, like Campylobacteriosis, actually the impact of that is much greater in a country where there's not necessarily great access to healthcare and possibly poor sanitation and possibly transmission routes through backyard chickens or I don't know, even person-to-person transmission possibly, or even asymptomatic carriage among humans. All of these things are very important, and I'm really excited to see that we have lots of talks that are starting to think about the global Campylobacter problem, which of course still remains and is still really bad. Sorry, Lisa.
Yeah, I think you're right there. I think we have been in the past a little bit Europe, America centric without giving consideration to our colleagues in those low and middle income countries. So it's really good to see that we're sharing that expertise and working with them and shifting that over. So Ben, I think you have the next question.
I was going to try and riff on some of the things that people have said there. So Sam's mentioned the variation within Jejuni and all these Campylobacter species. This is one of the challenges we've seen with making vaccines for chicken in the chicken industry, and then bringing that to the low middle income countries. Do you expect to see these same challenges if you were trying to make a vaccine, say in Africa, to try and reduce the burden of Campylobacter carriage in the Gambia or Zimbabwe? Do you think it would be a feasible project to make a vaccine to help reduce this? Shall I go to Brendan with this one? I know you've done some work in Africa.
Yeah, I'm quite happy to answer that. We have been working low and middle countries in Africa, the Gambia, for example, over a decade, and also, and we're part of a huge consortium that's looking in Bangladesh, India, Pakistan, Sri Lanka, and Vietnam. And I'm happy to comment on that later. But regarding a vaccine, and Christian might back me up here, we do believe that glycoconjugate vaccines have enormous advantages, and the WHO recommended for humans. And I know that humans and chickens are different, but certainly glycoconjugate vaccines work for mammalian species, and we'd be amiss not to really test these out in chickens. So obviously, given our background, we'd focus on the heptasaccharide. It's a nice 7-sugar structure. And the reasons for choosing that is that it's absolutely conserved amongst all Campylobacter jejuni and coli, and actually Campylobacter hepaticus as well. But it's also a good candidate because it modifies more than 100 proteins. So these are glycoproteins, and not always on the surface of Campylobacter, but you would have thought that that's a really good target for vaccinating around the world. So we, and I'm sure Christine as well, have been coupling this heptasaccharide to various carrier proteins, and we're testing these out in various poultry, but we're also actually testing them out. The difficulty with vaccines for Campylobacter is quite obvious. They don't cause overt disease in chickens. And we don't understand enough about the avian immune system. So our approach actually is we've made these vaccine candidates that they're made in Salmonella or avian pathogenic strains that are used by the industry. But we're also not just testing them in Campylobacter jejuni models, but also Campylobacter hepaticus. So we would want to see whether this would stop spotty liver disease. So that's more of a systemic infection where you'd expect a Campylobacter. So I think we'll find some quite good answers there as to whether you can make a Campylobacter glycoconjugate vaccine. And I think it's really important that we try, and there's no doubt about the need for such a vaccine. But who will pay for it? How much it would cost? These are all separate issues. We expect them to be low cost using the platform technologies that we use and Christine uses as well. But another point for discussion is I've had many discussions, Food Standards Agency and government ministers, they all want it, but they're not willing to perhaps pay farmers or industry to have these vaccines. But it would be cost effective for the UK or US economy because of the loss, not just preventing disease in humans, but the loss of income for people being off work. So there's a good cost economic model there for Campylobacter vaccine, and I think it's something that we'll try to work out one way or the other. But at the moment, with genomic information, we can pick a conserved protein or a conserved glycan, and there is a really good candidate for that. So I don't think the issue of strains being different in different countries, which they will be due to lipooligosaccharide, but not necessarily for the glycan. So I think I've said enough. I'll give Christine an opportunity to discuss this.
Ben, do you want me to go next?
Yeah, go on, Christine.
So I obviously agree with everything Brendan said. We also target that conserved hepatosaccharide, that we also don't see variation. And we've also been looking at Campylobacter structures in other countries. So we paired up with the Global Enteric Multi-Center Study Group, GEMS, and looked at the Campylobacters that were isolated in those conditions. And actually, surprisingly, and this is a paper that's going to be coming out soon, we do see one variant of the hepatosaccharide where Campylobacter instead makes a hexasaccharide, so it loses the glucose, and it's due to a repeat in the glucosyl transferase. So we're actually exploring that in a little more detail, but it still points to what Brendan has said, that core structure is similar with or without that glucose, and we think it's a good target. As Brendan mentioned, it's been difficult to make a vaccine against this structure, or any vaccine in chickens for that matter, because as he mentioned, there isn't that overt immune response, even though we do see spread and a little bit of consequences to the chickens. We really don't have the immune system on our side. And so what we've been finding recently is that the birds that clear it, they clear it. And, I saw in the chat, there was a question about how much does the microbiome come into effect. And we think that it might be playing a role there. But the birds that don't clear the Campy, very quickly, you will see the numbers come right back up to the numbers you started from. So we've been calling these responder and non-responder chickens. And so we've been exploring that in a lot more detail. But we've also been looking at opportunities for human vaccination, because as Brendan mentioned, and Sam too, that in low and middle income countries, there's a lot more serious consequences to children that are infected. So there's these links to growth stunting. There's higher rates for development of Guillain-Barré syndrome, which we haven't discussed much at this meeting, but we have a big DoD grant to investigate the consequences of GBS development, both in the US military as well as in Bangladesh populations. And so in those studies, they're not finding, at least from what I'm aware of, and Ari would have been a good person to comment on this because he's been recently funded by the Gates Foundation to do more source attribution studies, but there isn't a clear link with chickens as there is in North America or Europe or the UK. And so that's why they're looking at potentially immunizing infants instead of looking at clearing in poultry. But I think there are definitely opportunities and targets, even though Campylobacter is so variable. And Arnoud mentioned it in the chat, and I meant to call that out, that not only is there phase variation in Campy, but also in its bacteriophages. And that was one of the highlights for me at this meeting, was Martine's talk and the Cell Reports paper that they published recently too.
We moved, ask Sam to comment on some of the source attribution or the differences in source attribution we think we're seeing in these LMICs.
Yeah, well, I mean, you could comment as well as me, Ben, but yeah, we started to think about the population structure that we find in different parts of the world. And of course, when you look in the UK and France and Germany and the US and Canada, we see the same clonal complexes. I'll still use that terminology, but basically the same lineages are found. But as we start to look in other places, we start to notice big differences. So for example, in Southeast Asia, we've noticed that there are, well, correct me if I'm wrong here, Ben, but a major 20% of all disease in this country, in Britain, is caused by ST21 clonal complex, okay? And when we look in Southeast Asia, we don't even see that clonal complex. We see lots more C. coli. We see much more AMR. You know, and also in Africa, we've seen,
and Peru as well.
And Peru, absolutely, Ben. Thanks. Yeah, we've seen new lineages, new strains, new branches on the tree that just don't really seem to come up in the UK. So there's obviously lots out there that we don't know about. There's global spread, and that's very important here. We know that there's global trade in livestock, there's global movement of people. So it's no surprise that there's global lineages of Campylobacter. We could have a massive long debate about that, I think. But anyway, let's perhaps not today. But also there are local variations, and I think we need to understand those local variations. But I'm really interested in hearing about the vaccine stuff. I mean, that's very interesting. And Guillain-Barré, I didn't know that was worse in Africa. So the question, but it's coming from my old MLST background. The question I would ask is Guillain-Barré syndrome in Africa still caused principally by the ST22 clonal complex? But I don't expect anybody to be able to answer that because it's a very sort of nerdy question. But that's the question that immediately jumped into my head.
Yeah. What I mentioned was Bangladesh, not Africa.
Oh, Bangladesh. Okay.
Incidence. But what's even more interesting, and I don't know if any of the panel members, and I want to give Arnoud a chance to talk too, is this recent outbreak in Peru of very high rates of GBS associated with Campylobacters. And if anyone's really looking at those isolates to see why there was such a large outbreak, so they had hundreds of people develop GBS within a very short period of time in 2019, just before the pandemic. And they linked it to certain Campylobacter isolates, but I haven't heard much more about it.
Yeah, it's all the same ST.
Yeah. I could comment that in China, certainly rural parts, that they empty the hospitals almost because they get Guillain-Barré influx seasonally. And this is going back 20 years ago. So, we actually had a lecturer from the local university, work in our labs, and they were saying it was true that this was a seasonal thing that they found. And Martin Blazer looked at that as well in the past. And actually in South Africa as well, there've been quite a few clusters of Guillain-Barré syndrome, Miller-Fisher syndrome.
So they were all the same ST, those South Africa ones, the China ones, and the Peru ones that we've looked at, Sam.
Yeah. Well, okay. I don't know the answer to that.
No, they are. I'm telling. Yeah, they are.
Okay.
I wanted to spring to Arnoud then. I'm trying to think of how to get that to Arnoud. So Arnoud, in your big AMR paper, did you look at any LMICs, or was it American?
There's not enough data to really do the comparisons. Again, the problem, as I said in my talk, is that you get a lot of bias in those isolates because they are outbreaks, they are specifically sequenced before that. I mean, with the vaccine one, for Sparks, you need a bit of disagreement. So one of the things that alternative to vaccination chickens, because if we only vaccinate chickens, then we still have a significant bovine, for instance, route of transmission. Because a lot of isolates have bovine origins and sequence types that we see. Human vaccination has its disadvantages, and I think it's a very touchy subject nowadays. So let's not go there. But one of the things that you see in C. coli is, C. coli, we see that there is a lot of C. coli isolates. But if you look at it, the ones that come from pigs and from bovines and so on, don't really have many human isolates in the same lineages. That's probably because of the slaughter procedure being so different, is that you don't get as much cross-contamination. So while vaccination is one thing, another option could, of course, be, can we actually change the procedures significantly so that we don't get the cross-contamination of meat? Because in C. coli, that clearly seems to have worked pretty well for the pork and for beef. It just doesn't work with chickens. So rather than vaccinating and getting a technological solution, are there ways that we can actually change the procedures that we don't need vaccination? And that will also not be dependent on changes in the genotypes or protein expressions or sugar expressions and so on. So I'm not completely convinced about the vaccine route as the best one. But I also see that changing all the factories is going to be a little bit of an issue.
I
We've
just
tried
wanted to
everything else for
highlight-
years.
Yeah.
Sorry,
But to
Christine.
highlight two of the talks that I had heard today, one was on, and I found that quite interesting, is that they actually found higher levels of Campylobacter jejuni and coli in bovine as well as pigs, and lower levels in chickens. But then after processing, and this was the talk by the Canadian representative actually, and she mentioned that in chickens then, those are where the highest levels were after the processing. So that seems like the obvious thing, right? Is biosecurity somehow changing the processing procedure? But then when we heard the industry talks, they specifically said that, you know, we've done all we can and we still have similar levels of Campy, and that treating once they're infected and contaminated there doesn't seem to be other things that could be done to improve the situation, and that's why they're looking for strategies to prevent colonization or contamination in the first place. So, you know, whether you agree or not, that was what was presented today. And so I'm obviously on the side of the vaccination, but I do agree that I think it needs to be a combination of both methods, right?
Who of my fellow panel members has actually been to a chicken plant and actually seen how it's done?
Mhm.
Because that's, if you see it, it is the speed, you know, all these scalding bath, everything that the shared and where all the birds go through, which is like, you know, it's heaven for cross-contamination and transfer. So if you only have a few contaminated birds or the ones in the batch before that, it spreads it everywhere. As I say, the problem is you would have to redesign the whole process. It's like not hanging your chickens upside down where they have this volcano effect when they release intestinal contents and they spray it on the other birds. But in a way, can we get, I know it's going to be a huge undertaking if you want to change that, but are they really looking at how to change the factory, the processing plant procedures or not? And I don't think there is a significant change there because it's probably financially unviable.
Well, that's it. You've hit the nail on the head there, Arnoud, haven't you? We've been around the block on this before, and obviously it's better if you can stop it getting into the sheds, and then it's better if you can vaccinate the birds so they can't get the disease, and then it's better if you can not contaminate them at slaughter. But it just costs money, doesn't it? I think. I mean, and also we know that if you freeze all of the meat, then the Campylobacter load goes down a massive amount. I mean, obviously it doesn't wipe it out, but it's a very easy way of reducing the amount of Campylobacter on the carcass. But British people for sure don't want you to freeze their chicken. So then we get into all those blast chillers and I don't know, all these different things that we've talked about them over the years.
I still remember the happy chickens.
The happy chickens. Yes, happy chickens. So anyway, I digressed a little bit there, but I was just saying it's really money, isn't it? I mean, if somebody firm in government, as has been proposed in the past, sets criteria for reduction in Campylobacter that actually have teeth, that people actually have to stick to, then I suspect that more would be done to reduce the amount of Campylobacter on meat, which in my opinion, it's criminal, the amount of Campylobacter that goes into kitchens every day. I don't think it's really acceptable, but there you go. I would say that.
I agree. It's a national disgrace. And I think Arnold it set off a few sparks there.
Yes.
There's a fire going in my ear here. And because I feel like we've been around this block many, many times, as Christine and Sam have mentioned. I would say at a farm level, if you do have an intervention of poultry, there is the chance that less would be cross-contaminated with other livestock. So, I think we know that's the main source. So it's the best place to start.
We need to be careful, though. It's not just that the producers or the processors need to change things. It is a combination where everybody has to contribute, because it's as well the consumer who should be less of a, well, let's not use any swear words, And I also not look at my own kitchen procedures, but because it's a do as I say, don't do as I do. But it is something that everybody will have to contribute to, so from the regulators to the processors to the consumers. Plus, we may not have to expect cheap chicken all the time. That's another thing that drives down. We need to match the price with what is expected from the product.
We could just chlorinate our chickens, couldn't we, like the Americans?
Yeah, I saw that Nabil is talking about chlorinated chickens. That is actually something that we really shouldn't go for, even though the problem is not, we've got to remember for everybody, the chlorination is not the problem itself, the chlorine. It's the fact that it hides bad procedures. So it's you only manage to reach the level, the minimum level of safety with chlorine means that your procedures and your welfare and everything is so bad that you otherwise wouldn't be able to reach it. So it hides bad practices more. It's not a chemical safety of the chlorination, because on salads and everything, it's fine. And it's just allowed in the EU regulations as well. So, but still, you won't be able to sell it here.
So we've got a question from the Discord from Alexandre. And he asks, considering we're talking about vaccines and we're targeting Campy, what other effects for the microbiome would you or might you expect if we did eradicate Campy? Shall I go back to Arnoud with that one? I'll start you off.
Sorry, I was just distracted for a moment. So somebody else either repeat the question or get somebody else first.
I'll move around my screen then. Let's go to Brandon then again.
I'll pass on to Christine because she's done microbiome work in chickens.
So specifically talking about our own vaccine, so we use E. coli, attenuated E. coli carrier that carries the Campylobacter and glycan. And so we've looked at the effect on the microbiome, and we don't see a significant change at all in the microbiome. So that was one of the concerns, is if you remove commensal/opportunists like Campylobacter from chickens, will you open the floodgates for a worse pathogen like Clostridium perfringens or another Campylobacter, for instance, like Hepaticus? And we didn't see differences. And so it seems like it is possible to remove the Campy without having a large effect on the microbiome.
I think some of the studies quite often when you have a potential pathogen in the microbiome, you always see Enterobacteria say increased at the higher concentrations. But of course, then you get a chicken and egg question, which is very, of course, especially with Campylobacter, an interesting one. Is that because of the Campylobacter, they are changing conditions so that the Enterobacteria say can grow better, or are actually the increased levels of Enterobacteria say creating conditions where Campylobacter can have higher levels, or both ways?
Great, thank you. So we've sort of been alluding to this as we've been going along, and on the panel we have many, if we combine all of your experience, then we have many decades of research into Campylobacter, and yet we haven't managed to eradicate it in the higher income countries. Therefore, what can we do to help our lower middle income countries? What experience can we share? Is there any approaches that we could take forward that would be effective in these countries, given all of our knowledge and expertise that has come from all of this research? So I'm going to put that one to Sam first of all, please.
Okay, great. So I can certainly start off. So one thing that we've got some money. We're doing a project in Africa where we're modeling. First of all, we're doing source attribution. So working out the relative contribution of different reservoir hosts to human infection. I'm sure most people know stuff about that. But then what we can use are modeling approaches to assign risk to different behaviors. Again, it's standard epidemiology that everybody knows about and has for years. But in a low and middle income country setting, that might be very important. We haven't finished it yet, and I'm sure other people are working on similar things. But for example, keeping chickens in your backyard might be a terrible idea. I mean, we know that chickens carry a lot of Campylobacter, and Ben and I were in Africa just before the pandemic, and there were chickens walking around people's backyards next to the thing where you pour the water, the water pump, and other things. You know, we can all imagine this sort of thing. So there could be simple intervention. So my response there, just to keep it relatively succinct, is that it might be easier in low and middle income countries in the first instance, because actually killing a chicken by hand or a goat and butchering it yourself and hanging it and then selling it in a market, I'm thinking about a kind of poultry processing in low and middle income setting. That might be safer in terms of meat contamination. So the route to human infection might not be contaminated poultry meat that comes out of a little polystyrene tray in the supermarket. It might be, you know, just large amounts of feces getting into the potable water supply. So different questions in LMICs. Some of them might be easier to solve, but obviously money, you know, is a thing always when you're trying to help people with these sort of problems.
If only there was an infinite pot of money that we could use. So Arnoud, do you have any opinions on what we could move to do to help with the low and middle income countries situation, please?
Well, the best way, of course, always is to make sure that they're not lower and middle income anymore, that they go to higher income countries. That comes with other problems.
That might be easier said than done.
But I mean, I think what Sam is saying about the direct contact, I mean, we have a different kind of campylobacteriosis in the, you know, especially in the Western world, where it's much more foodborne from supermarkets, and not so much what I usually call the rural way of getting Campylobacter, which is more animal contacts ways. And that one will be more into the lower and middle income countries, as Sam already says. So we may actually have to look at what works in preventing this kind of more contact way of contamination and infection, rather than at the food transmission one, where it's more about the processing and the whole food chain. And then on the other hand, we need to be a bit careful about the contact route as well, because if we think about how many of Europeans, especially British, go around with their pets and what they allow their pets to do to them, and if we look at it, there is not as much disease transmission as you would expect from the animal contacts. That is definitely happening in many cases. So it may not be as bad as we think it is. So the transmission routes may just be more varied than they are in the higher income countries. And so that also means that you won't find an easy silver bullet that takes care of all of them.
Yeah, still looking for that magic silver bullet in the higher income countries. Brendan, carry on.
Well, exactly. We haven't solved it in the higher income countries. So, and actually to a large extent, similar to what Christine found in their studies, Campylobacter isn't so common in poultry in low and middle countries. And we've looked at many countries now in Southeast Asia, South Asia, and Africa. So the routes of transmission are more diverse. But one of the things I did want to discuss with the panel is this old concept that a disease is different in low and middle countries, where you have a watery diarrhea, whereas in the West we have a bloody diarrhea. Is that really true? I mean, what is the true evidence for that? And as countries become low from middle income to higher income countries, would that disappear? So in terms of solution, it's difficult. You need a multifaceted approach. I don't know a short answer, but the Poultry Hub, the global, the GCRF funded group that we're in, which is a £19.6 million grant from the government. Only 1.5 million is actually looking at the bug itself. Most of it is social interventions and look at markets and the chain. And they're trying to compare countries. One it's a low income, one it's middle income, and I guess Sri Lanka, which is more higher income, that has a lot more poultry production. A direct comparison between all three. So really, that the emphasis of that study is more towards social interventions that are cheaper, because it does come down to cost in the end.
Well, can I just say a quick thing? You sort of mentioned there, Brendan. One thing, I mean, I don't have any great knowledge of beyond Campylobacter, but it's clear that Campylobacter is underreported a lot in LMICs. And actually, when it's found, so I'm thinking now about the GEM study as well as some other studies that Christine mentioned. You know, when you get Campylobacter confirmed through testing the feces, you often get other bugs. That's the thing I'm trying to say. So you know, maybe you can't tell whether it's causing bloody diarrhea or watery diarrhea, because when you get Campylobacter in Africa or whatever, you often get salmonella as well, and sometimes you get some sort of bad E. coli or something.
Cholera, yeah.
You know, or cholera. Exactly. There you go. So all sorts of other nasties. So I wonder if that would be the first thing to rule out.
So let's see if I could remember all the things that have been said. So I have a few points about all of them. One is I do think it will be more difficult even to treat people in LMICs, because as Sam mentioned, poultry is one source, but there's so many other sources. And I'm seeing that from all the studies that are being published, that are being funded by Gates and others. That's why we've started thinking about whether it's appropriate to immunize the children instead from that perspective. And as Sam mentioned, the mallet study particularly is one where they showed that out of children that were under 1, 85% of them were Campy stool positive. And so you do see a super high level. And when we worked with the GEMS group, we asked for their table of Campy and co-infections. And I struggled to find a single child that had only Campy. So they were infected, not just with 1, sometimes 2, sometimes 3 enteropathogens. So I think that might also contribute to, as Brendan was mentioning, the watery versus the bloody diarrhea. And then as Arnoud mentioned, that happened also in the US, this contact with the pets. So we were having, for the last year or two, a big multi-state outbreak of puppies carrying antimicrobial resistant Campylobacters that they were actually struggling to treat because of all the resistances that they had. And it seems to have been linked, talking to my CDC colleagues here close to me in Atlanta, linked to the transport vehicles that are commonly used between different distributors that take the puppies to different pet shops. And it goes back to the mothers being treated with all sorts of very high doses of antibiotics. And so that's what they thought the scenario was due to. But it is also due to other sources. But definitely, I think in the developed countries, it seems more linked to the poultry, and Sam's group and others have done that through a lot of the source attribution studies. But elsewhere, it is going to be a struggle, I think, to treat.
There's a follow-up question on that from the floor, which is for those members of the panel that do work in abroad in these low and middle income countries, amongst others, how do you work with the local cultural practices and try to implement measures put in place or controls put in place in other settings? So I guess how challenging are the local practices to potential interventions that could be introduced? So I don't know if anybody has a strong viewpoint on that and would like to kick us off, because I know a lot of you have experience working with different countries. So who would like to start us?
Well, within the poultry hub, as I said, they've hired people specifically to go to these countries, and obviously the diverse ethnic groups and religions and practices, and basically I guess the sociologists have gone in first before we've actually gone in with our agar plates, if you like. So that was actually the start of the study, to understand and engage with the local populations, you know, before we've even mentioned Campylobacter. So yeah, it is very important. you have to engage with the local population before you can really do anything.
But that requires a setup like the MRC Gambia, where you actually have the institution there and also the people working there that engage with the local population. It's very often it's been people from Western countries going over to the countries and then taking materials back with them. And that is not, it's been shown so many times, that is not the way to go and not the way to get engagement.
Well, you have to capacity build within the country, and certainly all of what we do, right down to the sequencing. And we had a brilliant talk from the group at the Quadram Institute, with Bilal, where they've actually done MinION sequencing, and we had people from Bangladesh presenting at the meeting, where showing that they can do that themselves. And surely that's the best way.
Yeah.
The GEM studies and the MALED studies have both been really good at doing that, haven't they? Driving local infrastructure development so the local researchers can do it themselves.
Where's that voice coming from?
It's from me.
Who's me?
Ben.
Oh, right. Okay. Sorry, can't see on my screen.
Of course, we could joke about with Brexit, how soon is Britain going to be in LMIC, but let's not go there.
So we're talking about eradicating Campy and LMICs. We think we've done a pretty poor job in the UK and America eradicating it from our poultry industry, but Scandinavia and New Zealand have done a pretty good job at eradicating Campy. Why do you think that might be the case?
Well, New Zealand hasn't. It has reduced it because it was extremely high, and it has reduced it to more normal levels for Western countries, but it's most definitely has not eradicated it. I can tell you that as somebody who's on their New Zealand Food Safety Research Centre International Advisory Panel, it's still a big problem there. So,
it's a good question though. I mean, if you just take Scandinavia, then
yeah.
But the obvious answer, of course, is that it's cold there. I mean, is that a sufficient answer? I mean, this isn't the first time we've thought about this, is it? Because I remember, was it Lisa? Anyway, different people from Northern European groups. I remember them showing that their Campy rates are much lower, and I'm almost feeling a bit ashamed as a British person that my industry's rubbish and theirs is excellent. But I can't help thinking it will be factors associated with production, and also I suspect that the scale of production, certainly in the UK, will be higher probably than as a major exporter. But I don't know that. That's an assumption. Anybody else got better ideas?
I saw a note from Anne saying it's cold in Canada too. But Anne, if I remember
course.
your talk, you actually did show there was a decline. Wasn't there also in the Campy numbers over time?
I'm not sure they can unmute themselves.
Yes,
this
I was
session.
looking in the chat to see if she replied, but that
But
could
I think
be a
I think it
variable.
is this. It's for the Scandinavian countries. It's scale. It's also I think from what I've seen from this is they got a lot more environmental isolates, for instance. So it's also where you look for. But because of scale, because of distances. I mean, with COVID, we've seen the same. Is that the numbers of infections were a lot lower, and that's just because it's a different type of setup than in the northern countries, the Nordic countries, than there is in the sort of West and South European countries. So I think that will play a role as well. And as I say, the climate, all those kind of things.
I think the public are more aware in Scandinavia. I think it was in Denmark, they put health warnings on chickens. So all interventions like that, and combined maybe with cold weather and that. But yeah, certainly I feel like the Scandinavian population have been better informed about Campylobacter.
Let's all follow the Scandinavian example then.
I think that's an excellent suggestion.
That it sounded like Frida.
I know, I'm not sure whether I'm allowed to talk.
Oh, it was Frida. Yeah,
Go
brilliant.
for it, Frida.
I think you are. Of course you are.
There are some there are some Scandinavian dishes that I won't touch, like the fermented fish stuff.
We're going completely off target here. So, I mean, they take biosecurity extremely serious in the Scandinavian countries. They go and speak to the processing plants. They've managed to, if they get a spike in human disease, they'll go and find out what sequence type it is. Then they'll go and find out what processor is doing it, and then they'll speak to the processing plant and the farm. And, but you can only do that when you don't have too much in the first place. In the UK, we wouldn't know where to start. We've got 10,000 SNP clusters, where should we start?
Okay. Ingrid here.
Hi, Ingrid.
Hi, I'm from Sweden. We actually have less than 5% of Campylobacter positive chicken last year, and we have about the same this year. I think that one thing is that there are quite a few producers. It's under 110 producers. And it's only about 10 of them that hit really high levels of Campylobacter. And it's easier to just visit 10 or 15 producers and say, you should do this and this and this, and then we should lower levels, compared to other countries that have quite a higher numbers of producers. So because we're going, and furthermore, we are quite really good biosecurities, and if they actually deliver chickens with Campylobacter, the producers get less paid, and you can do quite a lot of money.
That's also what the New Zealand one. Of course, New Zealand had the advantage that they were pretty much self-sufficient, so they didn't import any chicken, but they really only have 3 big chicken producers. And there is actually a financial penalty, very strong financial penalty, if you're several times over the limit of what is allowed for Campylobacter. You're only allowed to sell it as frozen meat and not as fresh meat anymore, which means it halves the value. So if that happens a few times, you quickly go bankrupt if you're not careful.
Yeah.
So there is a very strong incentive on changing. And for instance, one of the things when I visited there, which they told me had made a really big difference in their levels of Campylobacter, was when they bought a new eviscerator, where they could change the, well, that's easy way to say, the size of the hand that takes out the intestinal organs, depending on the age of the chicken. So if it was a 30-day-old chicken, it's smaller. So you needed a smaller hand, so you got less rupturing of the internal organs, and that meant less cross-contamination. And that was the biggest progress they made, is buying a new eviscerator, a much better one. But again, the cost of that was very significant, of course, but it paid itself back by getting much lower Campylobacter levels.
Yeah. In Sweden, we slaughter most of the chicken. If those who do the finning, slaughter at day 28, and then they have the head slaughter at age of 35 days. I think that's a little bit less than quite a lot of other countries in Europe.
Can I just ask a question, Ingrid?
Yeah,
Did you just mention thinning there?
Yeah.
Okay, so that was what I was going to ask, is do they do thinning?
Yes, but it's only one slaughterhouse in Sweden that use thinning, and that's the biggest one.
And is that the one with the most Campylobacter?
Yes.
Okay, well, that's quite interesting, isn't it? For those that are not familiar, thinning is a practice of removing a proportion of the birds before they're fully mature, because then the remaining birds grow to fill that space, and you end up with extra yield.
Yes, especially if you have a problem with to have clean transport crates, because that's a big problem in Sweden to clean them properly. I don't think there is a good cleaning routine for transport crates anywhere.
Yeah, it's also that there were teams of finning teams going around to different farms, which of course is the worst practice if you think about biosecurity. If those that go into the birds and take them out and then go to another farm, despite all the cleaning and everything, the risk of cross-contamination, of course, increases the more visits there are of the different teams. So all these kind of things play a role, and the no finning policies that the UK, Mrs. Marks and Spencer, started doing should pay off at some point.
And the slaughterhouses don't have loads of crates, so they use the same crates at least twice a day, and you don't have enough time to clean them and dry them in between.
Great, thank you very much, and thank you for the additional input there from the floor. I am going to change tack slightly now, if that's okay, and you don't mind. So we've heard quite a lot, not just from this meeting, but in the past, about how chickens are a changed host. So a chicken now is not what it was 5 years ago, 10 years ago, 50 years ago, and they're very different. So I would quite like to know from the panel how Campylobacter has changed to with that host. So basically their views on host adaptation in Campylobacter. So I don't know if there's somebody who would like to kick us off. Sam's nodding his head. So I think I'm going to put it to Sam first.
Well, I like thinking about this stuff because well, everybody knows zoonoses, host adaptation is massive. I mean, if we didn't know it before, we really know it after this awful virus suddenly adapted to humans. So, you know, switching hosts, changing with your host is a massive thing. How Campylobacter reacts to changing chickens over the years is difficult for me to answer. I suspect that you know, the way that chickens are bred to grow very, very fast is disrupting their immune response somehow. I would love to have more information about how Campylobacter colonizes, you know, ancestral chicken breeds and what immune response is elicited. But I suspect that Brendan or Christine could speak to that better than me. But I think that we are learning more than ever before about host adaptation and how bacteria evolved to colonize new niches. And I think this is essential. You know, I think this is an area of Campylobacter research that goes way beyond the boundaries of this organism. And we can really start thinking about what happens when a Campy or an E. coli or a Salmonella moves from one host into another. What are the genomic changes? How do they benefit? And you know, risk of sounding a bit of a sort of techie kind of a nerd, you know, this genomics revolution that we've lived through and continues has allowed us to think all of this stuff. And now people typically will think about multiple genes that are associated with host adaptation. They'll think about the source of given genes that are moving through horizontal gene transfer. And actually for me, maybe I should have said this in the first bit where you say, where do you think the field's going? But I suppose this is beyond Campylobacter for me. It's now going to be thinking about how genes interact within a genome. So this might be going a bit into, I don't know, not what we usually talk about at Campy UK, but for me, I'm really interested in how genes interact. So if we have a given chicken-associated gene in the genome, does that mean that other genes are going to change to sort of match that? And the applied benefits of this are enormous. It helps us to think about source attribution, knowing where particular strains came from. We can think about the putative function of genes that are clearly important in surviving in a given host or niche. We can think about targets for vaccines or for other genome therapies that might help us to remove some of these bugs. So kind of ran down a few routes there, Lisa, didn't I? But that's my starter for 10 on host adaptation. But how they adapt to chickens. I mean, obviously, I'll end by saying this. What we really need to try and do is produce a chicken that grows to its full weight. This is purely from an economic point of view. It's actually probably ethically terrible. But we need to produce a chicken that grows to its full weight before the maternal antibody is lost. How about that? Can we do that?
5 days, Sam, is that what we're saying?
If we could. Like, what is it? It's 32 now. It was 75 50 years ago. How long till we can get it down to 5?
15.
I think actually
I don't think the chickens changing makes any difference. Campylobacter jejuni is on this planet because it loves colonizing the crop of avians. At 42 degrees, its optimum body temperature, and it absolutely loves it. It stuffs in the crop. So it's on this planet. It probably was here before humans and pterodactyls or whatever. So I think that a slight evolution of chickens over a few years isn't going to make much difference. But I will say that Sam has done some elegant work and host adaptations, such as the bovine version. But yeah, honestly, you know, we know it's there in all birds, seagulls or whatever, geese. So I don't think chickens changing over a short period of time is going to make any difference. That's my view.
I think we had a change, though. I think you know, Ben already said about the sequence types you find in other countries. And what we may actually have there is that within the Western countries, because we've upped our chicken production, and so we may actually introduce the bottleneck towards certain strains, which is the ones that we sequence all the time, and we have way too many of already. But in the lower middle income countries, a lot of them will be more like the old-fashioned chickens. I put a picture of the 1957 to 2005 on the chat. And actually, we should probably look more in those chickens, and maybe in Peru, Bangladesh, and so on. Those chickens will not be like the 2005 chickens. So, and that may partially explain maybe why you get such difference between those countries, and why we see so much of ST21, 353, 464, all those kind of genotypes which are really abundant in chickens, 257 and so on. So, rather than doing more in Western countries, I'm going to quote Martin Maiden on that one. We should stop sequencing any more Campylobacter until we've analyzed what we already got. And actually, if we start doing anything more, we should do it elsewhere. Again, sparks required. So, but I think we really are overdoing it in the Western countries and getting way too much information. And we should invest more in getting a better understanding of going back a little bit and going to a more worldwide view of what is out there.
I agree. And fewer clinical and chicken samples. Let's figure out where it came from in the wild, you know.
And remember, I'm not sequencing, so who else is sequencing? Stop it.
So maybe just to add on a few of the points, see if I can remember all of them. I like this idea of looking in some of the heritage lineages of the chickens to see, is there any differences compared to now? And what specifically I'm thinking about is this spread to other organs. It wasn't reported before. Is it because we didn't look so hard, or is it because there wasn't really any spreading? So that'd be very curious. There were some talks also at this meeting that kind of grabbed my attention, and in the chat, Chris Bayless mentioned that the phase variable genes in Campylobacter don't really change so much at the beginning of a chicken's life cycle, but if you go out to 52 days, then you start seeing distinct phenotypes that are potentially being selected for. And then during the industry talk, we also heard that the backyard birds, the free-range chickens and organic chickens that are kept longer kind of grow out of their Campylobacter, I think was the term used. And I thought that was actually quite interesting too. And then just one personal observation, which we're following up on, is we've been playing around with generating germ-free chickens and introducing the Zika later. And what ends up happening is you get an underdeveloped intestinal tract, so you don't get as many villi and divots as you would expect in a regular chicken, and we see less colonization of Campylobacter. So maybe by making these chickens grow so well, you do somehow make them more susceptible to Campy colonization. But this isn't published yet. This is something very, very new that we're following up on. But I think you raised a really interesting point, and all the speakers, I thought, made very interesting points too.
Thank you. And Christine, I look forward to seeing the studies once they're published, because it sounds absolutely fascinating.
Can I say one more thing on this one quickly, Lisa? I promise.
Of course you can, Sam.
I've always been really enthusiastic to do what I would call a host pathogen genome-wide association study. So if we can find chickens that are quite well resistant or have lower Campy load, and then we've got some that have high Campy load, because of all the wonderful genomes and the wonderful analysis tools we have now, we can say to ourselves, okay, which genes in the chickens make them resistant and which genes in the pathogens make them pathogenic. I think that would be a fabulous thing to do. I just wanted to say that. Anyway, there we go.
Thank you. Okay, so I think we had one question that was on the Discord, which is slightly, we haven't put it in yet because it didn't really fit in with our topic of conversation. But as we're near into the end, it's the perfect time to ask it. So I'm just scrolling back to find the question so that I get the wording right. So the question is there any link of Campylobacteriosis and modification of the gut-brain axis? So I don't know if anybody's got any thoughts on that one, but I thought as it was, I thought it'd be a good final question if anyone's got any thoughts for this one.
Well, it clearly must make people crazy if you look at the panelists that are on this discussion right now.
I haven't had Campylobacter as far as I'm aware, so I can't talk from personal experience.
I suppose my response to that would be, aren't those sort of things usually associated with long-term carriage of a pathogen? That's a bit of that's boring compared to what Christine and Arnoud said, but it's a sort of, isn't that the case? So if you had it for ages, maybe you could say it makes you risk averse or something, whatever they say about these sort of things. But also,
I don't think the microbiota will change, as Sam says, significantly from a Campylobacter infection, even though a strong immune response for a short term can have these effects. And there are stories about linking with irritable bowel syndrome, inflammatory bowel syndrome with Campylobacter infections or other enteric infections, heavy ones, or the associated antibiotic usage that can cause changes in the microbiota. The problem lies, of course, is that that's only talking about the microbiota and the intestinal syndromes that you can come from it. Effects on the gut-brain axis and so on will be a lot more difficult because they tend to occur over much longer time and much more subtly. So I think you're going to have to go and have to find people who get at least 5 Campy infections per year to and don't learn from it, and that may be the effect on their gut-brain axis.
I've got another quick thing. Am I leaping in front of some urgent? Anyway, I just thought to myself then, perhaps our question, I don't have Discord, so I can't actually read the question, but maybe we shouldn't be thinking about people. Maybe the question is, does it affect chicken behavior or cow behavior or something like that? And there I could certainly see potential. And Fran Collis, who gave a talk here, but I'm still catching up, but she did some work recording the movements of chickens, I think, videoed it and showed, because the obvious question is, how do you tell if a chicken's ill? I mean, we've all said, or certainly Brendan said this earlier, that we still don't know if there's still debate about the extent to which Campylobacter is a chicken pathogen rather than a commensal organism. I think we all know that. But and one of the reasons it's still a debate is you can't ask a chicken, if it's got tummy ache, because you can ask it, but you won't get much of a reply. And one way of trying to figure that out, of course, is by observing the behavior of these animals when they're infected and when they're not, and seeing how that changes. And I mean, I've seen chickens with what I would call diarrhea, but I think you've got to do quantitative analysis of this. So maybe it changes the behavior of hosts like that.
Time for the chicken psychologist.
Yeah.
And for those of you that are interested, Fran has just shared the link to that paper that Sam's just spoke about in the Discord. So it is there. So thank you very much to the panelists. We're going to move to wrapping up the conference. There is still lots of interaction on Discord, lots of comments. Your responses to the questions have sparked lots of debate. So do please take a look at those if you have time. So we've now reached the end of the formal programme for Campy UK Goes Global 2021. It's been a really exciting meeting, and it just goes to show that we really do have a strong scientific Campylobacter community across the globe. Within each one of our 4 scientific sessions and our industry session, we have had approximately 100 participants on the Zoom calls and very engaging conversations on Discord. And this conference has been a success because of the people who took the time to submit all of the abstracts, agreed to give the talks, and have actively engaged with the sessions on Discord. So we'd like to thank all of the speakers that have contributed to our meeting. Also to the industry and the public health panelists. We've had 2 great sessions over the last 2 days on those. Of course, this meeting wouldn't have been possible without the help of a lot of people. So we need to just take a moment to thank all of those that have been working behind the scenes and on camera as well. So we'd like to, on behalf of the organising committee, just thank the excellent chairs that we have had during the sessions. We've had the session chairs out front, keeping everything on track, but also the moderators behind the scene. So to everyone that has chaired a session, so that's Caddy, Desi, Cynthia, Janie, Andrew, Evangelos, Ricardo, Lisa and Roberto, we would like to say thank you very much for taking that time. We'd also like to just say a quick thank you to the team at the Quadrum Institute Bioscience who have hosted the Zoom sessions for us, been giving us advice, handling social media, organising the upload of the talks to YouTube. We really couldn't have done it without them. The talks have been uploaded in a very prompt manner. So to Andrew, Andy, Becky and Laura, we would like to say thank you very much. Of course, we've just had the excited site and plenary session, and we very much appreciate the guys still on screen who give time this afternoon to actively take part in the session. So thank you very much to Arnoud, Brendan, Christine and Sam. And Sam, we send you very best wishes for a speedy recovery, and we hope that your football career will be back on track very soon.
Thank you.
Finally, to the organising committee, so my fellow committee members, Ozan, Ben, Nicole and Nabil, what can I say? It's been hard work, but a lot of great fun putting this together with you all. So thank you very much for making Campy UK Goes Global 2021 such a success. And with that in mind, we look forward to handing over the baton to whoever would like to organise the next Campy UK. You know, you don't have to take it global, you can keep it as a Campy UK should you want to. And to help with this, it would be really great if participants of this meeting could take a few moments just to fill out the feedback form that is on the website. We want your feedback, we want to know what's gone well, what's not gone so well, so that we can pass that on to the next organisers. Ben.
And so the last thing for me to do is that the Microbiology Society and Microbial Genomics Journal has kindly given us some money to award to the best lightning talks. And I'm going to hand over to Sam to announce who's won those 2 prizes.
Okay, thank you, Ben. And thanks to all the organisers again. I think I know it's a lot of effort, and I know we're all really grateful as a community. So yeah, hence the background, you know, so I'm deputy editor-in-chief now of this journal. So I have to say, I encourage you to submit here. We're very interested in Campylobacter. I can commission papers and all this sort of thing. So if anybody's interested, just email. But yes, I have the privilege of awarding a small sort of gift prize for the best 2 talks of £75, again, sponsored by Microbiology Society and MGen. There were lots of good talks. I've been catching up. I'm still not quite caught up, but there are some that I wanted to mention that are runners-up to the prize. So I just wanted to mention Samuel Bloomfield, Evangelos Morkas, Heather Chick, Zhenfeng Cao, sorry about the pronunciation, Blage Jug, Agustin Conessa, and the 2 winners of the prize this year are Gillian Carney and Nafiz Assan, okay, for their work on fluoroquinolone resistance in an anaerobic environment and risk factors for pregnant women, respectively. So very well done, Gillian and Nafiz. Excellent talks, really thought-provoking, as well as rigorous and, very exciting stuff. So, virtual round of applause, and we will organize the prize to be sent to you forthwith. Okay, is that okay, Ben?
That's brilliant. Thanks, Sam. I think we're going to have a special issue as well in microbial genomics. So for those who presented unpublished work, it could be a good place to send your paper.
Yes.
And then I'll just close by saying, well, thanks to everybody, because every time that I attend a Campylobacter conference, it's always a great experience. It's such a friendly community, and there's been loads of engagements on the Discord, and Discord will remain active, so you can carry on chatting there and trying to build collaborations and further your research. I think next year there is a plan to have a CRO 2022 in person in China, but maybe Christine knows a bit more about that. And now this will be the formal end of the conference, but there will be a pub quiz in the pub on Discord for anybody that wants to come and join us. And there'll be lots of quiz questions about Campy, maybe. Okay, well, thank you. See you all soon.
Bye, everybody.
Bye-bye.
Nice to see you.
Bye. Thank you.
Thank you. Bye-bye.